Okay, so we're going to conclude our presentations for the day with the second round of lightning talks. Hopefully this will get everyone's heart pumping again a little bit, at least those people that have to present their work in 30 seconds. So to just refresh you on the format, there will be one slide per person. You have 30 seconds to summarize your research and to entice the audience to come to your poster afterwards. So we'll begin.
Hello, my name is Jeff Goldie, and I'm presenting on the Fullon QPCR platform. This is the microfluidic platform that was discussed earlier today by the single cell team. This platform was developed in collaboration with research science and structured science, and is a good example of various research teams working together to develop a standardized robust platform. Thank you.
Hi, I'm Kate Roll, and I will be presenting information on intrinsic signal imaging, or ISI. Basically creates functional maps for future targeting, such as two-photon work, viral injections, electrode placement. Dan Denman showed some of our work earlier today and how it can be used. If you think it would be useful to you or you want to learn how it works, stop by my poster, 2.2.
Hi, I'm Marina, and so a big goal of the Institute is to create a detailed catalog of visual responses, and not only the higher visual areas, but also or not only the primary visual cortex, but also higher visual areas. And so before organizing a big scale project like this, we have to do a pilot study to determine what the most efficient and effective stimulus is to continue on with. So please join me and Saskia de Vries at 2.3.
Hello, I'm Bethany Danskin. I work in neural coding. In this project, we take transgenic mice that express channel rhodopsin or archaeorhodopsin in cholinergic neurons and train them to the virtual foraging behavior task, in which they run on a wheel and discriminate between visual objects. Here they stop for the rewarded target object and run past a distractor object. We then perturb the behavioral performance with optogenetic inactivation or activation of the cholinergic system and record performance. Thank you.
Good afternoon, everyone. I took the request for a lightning slide literally, and what you see here is lightning crackling across the surface of the left hemisphere of the brain as a mouse runs on a wheel and is presented with visual stimuli. If you want to know how, or indeed why we do these experiments, stop by the poster.
Hi, I'm Refugio with the in vitro human cell types, and we make reporter lines on human embryonic stem cells. And today I'll be showcasing work that was done to characterize our double cortin reporter line, which is double cortin is expressed in maturing neurons and very important in neuronal migration. So if you want to see some more yellow neurons doing gymnastics, come and see poster 2.6. Thanks.
Good afternoon, everybody. My name is Nick D. The title of my poster is Development and Implementation of Histological Techniques for Morphological Characterization of Neuronal Cell Types in Cortex, which is basically just a longer and less fun way of saying more histology for morphology. Please stop by my poster for examples of techniques currently up and running at the Allen Institute, as well as platforms that we hope to develop in the near future. Thank you.
Hi, I'm Marina. I'm in Sean Olson's group, and my poster is about the work that we're doing to relate the responses of cortical neurons to the behavior of a mouse performing a visual task. So if you want to find out how this movie was created, what brain area this is, what task the mouse was performing, and a beginning look into how the responses relate to the behavior, come check out our poster 2.9.
I'm Tonya Daigle, the Manufacturing Process Engineering Group. And our display is the Experiencing Mouse Visual Foraging Project, loosely derived from inspiration from two great visionaries and based on a Simpson, Duffless, Fox episode. So we challenged the group to participate in the human foraging experiment with results posted for everyone to see. So let's get ready to rumble. Thank you.
Hello, I'm Fullon from technology team. What you're looking at is an image of gene expression in human glioblastoma characterized by ISH. Can we identify the boundaries between different regions, such as tumors and necrosis, and quantify the gene expression level in each of the regions? Also, can we develop a pipeline to search for the entire database for user-defined gene expression profiles? The answer is yes. If you know how we did it, please come to our poster number 11. Thank you.
Hi, my name is Ralph Puchalski. And why would anybody study glioblastoma? Because it's deadly, right? It affects 4 people or 3 people out of 100,000. And if you're diagnosed with GBM, you have about 4 months to live. If you opt out of treatment, if you elect treatment, you have about 15 months. Either way, the prognosis is very poor. So come to Rachel's poster to see what we're doing to change that.
Hi, my name is Yang Li. I'm from technology. What's that face? It's every one of us. It's the average face of the Allen Institute. We made it using the same technology we made our average mouse brain. So if you want to know more about this and many other amazing technologies, please stop by poster number 13. Thank you.
Hi, I'm Julie Nyhus, and this poster is going to describe the processes involved in the acquisition of both adult and fetal human brain tissue. So we're going to follow a post-mortem adult human brain from the medical examiner's office through case consent, case qualification, through the dissection, the sectioning, and the freezing that's done by the Institute's tissue procurement team, and ultimately the transport and intake of this really well-defined tissue at the Institute for use in multiple projects.
Hi, I'm Jeremy Miller, and I will be presenting a component of the Brainspan Atlas of Developing Human Brain, which focuses on mid-fetal human development. This atlas includes in situ hybridization of 46 genes, 2 reference atlases, and over 1,000 microarrays spanning microdissected sections from several hundred brain regions. If you want to know about how gene expression changes across different regions and layers of the brain and how expression in human differs from mouse, please stop by poster number 15. Thank you.
Hi, my name is Brian Lee, and we are presenting our efforts to acquire a transcriptional profile from electrophysiological recorded neurons. So please come by and check out our poster and hopefully provide some insight on how we can improve our methods.
Hi, I'm Tuk Nguyen. I work on a team led by Bosukotasik, and we are trying to identify cell types and also genetic markers for these cell types by analyzing the transcriptome of FACS-sorted single cells. We focused on single cells in the mouse primary visual cortex, and we've been able to identify known and also new cell types. So come by my poster to find out more.
Hi, I'm Trig Babakan in the Human Cell Types group, and as you can see, there's been a massive increase in brain size in the primate lineage and lengthening of cortical development. If we want to understand how evolution has shaped our brains, we need a high-resolution atlas of when and where genes are expressed in the developing brain. We now have this atlas in macaque monkey, and so if you want to know more about questions we're asking with that, come stop by poster 18.
Hi, I'm Doug Olorenshaw. I work with Sean Olson in the neural coding group, and we're ultimately interested in both monitoring and perturbing neural activity while animals are engaged in visually guided tasks. So please stop by my poster. I'll explain some work we've been doing on scaling up the basic visual foraging task as well as developing novel tasks. Thanks.
Hi, I'm Naveed Mastin with the Neurosurgery and Behavior team, and we have a quick video.
Oh, hi there. Welcome to the Neurosurgery and Behavior Surgical Suite.
Here in the surgical suite, we perform a variety of procedures, including the head post and craniotomy procedure, the head post for electrophysiology procedure, serotaxic injections, and the recover slip enterotomy procedure. If you come to the neurosurgery and behavior poster, we will take you on a journey from beginning to end and show you what it takes to become a neurosurgeon. So if you excuse me, I have some work to do.
I'm Ryland, and I work in the neural coding group with Jack Waters, and what I'm interested in is how the rest of the brain signals information about behavioral state up to the visual cortex. And so with that in mind, I'm interested in different neuromodulatory projections and imaging their activity during attention-related behaviors. So come to my poster to see how I'm going about doing this.
Hi, I'm Jun Zhang, working in Jack Waters Group, and traditional retinal topic mapping and visual area delineation usually requires hours of recordings. And in the past year, I'm working on recording wide field fluorescence imaging from GCAM mouse lines, and I'm able to do retinal topic mapping less than 20 minutes and automated visual area delineation. If you are interested in this, please come to my poster 22. Thank you.
Hi, I'm Linda Madison. I work with a small team in R&D to generate transgenic mouse lines designed to facilitate studies of neural circuitry. If you come to my poster today, I'll talk about our recent work to develop improved strategies for transgene expression to achieve both increased specificity and increased levels of expression. I'll also introduce a set of really exciting new lines we made recently that robustly express a variety of novel and highly effective genetic tools. So.
Hi, I'm Carla Hirakawa, and I work with Julie Harris and her team on the next step after the Mouse Connectivity Atlas project, which is next generation connectivity. We're working towards building a comprehensive structural connectome of the mouse visual system, and my poster will be showing some of the new viral and traditional tracers that we've been using to pilot this work. So come and see my poster to see what works, what we've tried, and how well it works. Thank you.
Do you love the Connectivity Atlas, but wish you could trace retinal projections to their targets in the brain? Well, now you can. Introducing the Allen Institute Retinal Projectome. We're bringing back over a dozen of your favorite Cree lines from the Connectivity Atlas, along with 15 newcomers, each carefully curated by our collaborators at Harvard. You'll marvel at intriguing differences in expression pattern between lines. And all of this comes in the stunning 3D you've come to expect, conveniently packaged with an image of the retina. The next generation of connectivity starts this October.
Wow, that's a tough act to follow. But anyway, I'm Tonya Daigle, and I work with Honghui and Ali, and we're developing viral methods for the next generation connectivity atlas. Come by my poster to learn about how we produce viruses in-house, what we're doing with them, and our analysis towards looking at neural circuitry studies. Yeah, thank you.
I'm Joshua Grimley, and I lead the group that makes transgenic human embryonic stem cells, and we use those as we work to study human cortical development.
Good afternoon. My name is Sherman Wang. We all like synergy here at this institute, so this afternoon I'll be presenting on what we do in the in vitro human cell types and how we synthesize our three main methods here, and differentiation, cell sorting, and single-cell transcriptomics.
Hi, I'm Julie Pendergraft. I work in the molecular biology department of structured science, and I'll be presenting on the Institute's internal genotyping service, and we'll be going through all the steps from sample submission through results reporting at poster number 29, the last one.